Alright, starting with section 3.2, looking at external structures for primarily here bacteria cells. The first major one is going to be the flagella. The flagella is a prokaryotic propeller in all sense of activity. Alright, so what does the flagella do? It propels the organism through its environment, so it's going to be used for bacterial locomotion. Alright, this flagella has three main parts, and I don't expect you to know the three main parts, but just so that you have an idea, it's going to have what's called the filament, and the filament is the tail-like portion that is on the exterior of the cell. Then it also has the hook, and I guess this is also on the exterior to an extent. The hook is going to move into the basal body, and the basal body is what sits in the cell wall of the bacteria, and it allows for communication between the interior of the cell, where the cell would have the energy to supply the fuel to move that filament. Alright, the hook moves with a 360-degree rotation. It's actually pretty cool. And this is actually an electron micrograph picture of the filament. And it's just, it looks like a machine, honestly. Blanking on the word there, ornate, alright, ornamental. It's just very cool. Alright, now flagella can have a variety of arrangements, and you see a variety of them here in figure 3.7. And this first one is going to be a single flagella. Alright, just one that can be called monotrichous, and I'm not going to require you to know these names, but just understand that there's a variety of ways that a bacteria cell can have or express their flagella. Alright, then B is lophotrichous, a small tuft or bunches of flagella at one end. Amphitrichous, alright, think about ambidextrous, someone who can write with both hands. And here you have a single flagella at each end of the cell, at each pole. Then peritrichous, flagella dispersed randomly across the surface of the cell. All of these allow it to move slightly different ways. Just because this guy has a bunch of flagella doesn't necessarily mean it moves better than this guy here with just one. Right? It's just a way that organism is made, how it has its genetic code, and what it looks like. So then the movement, some of these words are important. Bacteria move in response to chemical signals, and that's going to be called chemotaxis. And that is an important word for you to remember. Chemo is chemical, taxis, like taxi, is a movement. And what happens is that receptors bind extracellular molecules, so cells outside in the environment. The receptors are going to be on the surface of the cell somewhere. The molecules bind to that receptor on the surface, causes an interior signal, and then it starts that basal body movement to then move the hook, that then moves the flagella. Alright, so then there are different ways to classify the movement. There are the runs. Alright, some bacteria give you the runs, but this is specifically talking about the movement of the bacteria. Alright, runs are smooth, linear movement toward a stimulus. Alright, so the chemotaxis, the chemical, is going to be that stimulus. It's being drawn to a chemical, to something. And so it's just going to be a straight, kind of a straight beeline movement. The tumble, on the other hand, the flagellar rotation, reverses, and it causes the cell to stop and turn its direction. However, if there is the peritrichous flagella arrangement, then the tumbling is just very random. It's going to take a longer period of time for it to reverse itself, but even with the runs, all of those randomly placed flagella work together and still allow the bacteria to move forward. All right, then looking at two other types of prokaryotic appendages. All right, you have what are called fimbriae and you have pili. And there is a very specific reason why you have pink here for the words, and that is because typically you're going to find fimbriae and pili on gram-negative cells. All right, gram-negative cells, when you do a gram stain correctly, those cells are going to appear pink. So you just have a visual connection here. Gram-negative, pink, you find the fimbriae and the pili. The fimbriae are small hair-like projections. It's very difficult to see these, but there's a couple right in here. You can see a couple here through the shadow of the capsule here. All right, down here is a pretty good example of all of those small lines that are coming out. Those are short hair-like projections similar to the hair on your arms. Those are going to be used for attachment. Very important. Very important means of pathogenicity for a bacterium to be able to get into its environment or get into a location and then stick. All right, then the fimbriae are going to be used for attachment as well, and this is generally going to be attachment to other cells, but the main purpose is going to be to allow for genetic exchange between two bacteria cells. So what the pili is, and sometimes it's referred to as a sex pili or a conjugation pili, because it actually is a narrow tube. And what happens is that the pili is produced by one cell. Let's say this guy produces the pili. It extends to this cell, and then it attaches and creates a hole into this cell. This cell is then going to transfer some DNA material, and it's going to come off of a plasmid. It's not its normal chromosome material, its genetic material that is in the nucleoid region, but it's that small circular piece of DNA that usually has pathogenic genes, like the antibiotic resistance gene. And that's a means for bacteria to be able to spread its DNA, and it's going to do that through the pili. All right, the pili is going to be much longer. And generally a cell only has a couple of these, 1 to 2 to 3 pili per cell. So fimbriae, very numerous, just like the hairs on your body, on the surface of your body, but very short. Pili, quite long and very few. All right, then you have the glycocalyx. The glycocalyx is going to be a means for evasion, really. The glycocalyx is made up of sugars, right? Polysaccharides, so it kind of makes it sticky. There's also going to be proteins that are involved, and it's generally a mixture of those two, the polysaccharides and the proteins. The glycocalyx can vary in how thick it is. And there's two types of glycocalyx. First is the slime layer, and then there's a capsule. And the purpose of the glycocalyx is to avoid phagocytosis. Right? Phagocytosis is what's going to occur with your immune cells. The phagocytes will come in and identify that that bacterium is foreign, and it tries to eat it up. Well, even though sugar, these polysaccharides can be sticky in nature, the capsule or slime layer actually makes the bacteria slippery. And so it just slips away from the phagocyte and it goes on to be able to replicate and cause more damage to the host. All right, now back to that sticky side, it can be used to adhere to surfaces and create biofilm specifically. So here you have a surface, you have some bacteria cells that are going to come along, and they stick to the surface because it's going to stick to this coating here, and you see that coating right here. The bacterium, once it sticks, it can actually have different signals that will cause it to start producing that glycocalyx. And now you can see these cells nice and puffy because they're producing high amounts of the glycocalyx, and they can replicate. And these cells basically are protected against the immune system, so they can continue to replicate to high numbers. And then in terms of looking at a biofilm, you don't just have those original bacteria cells. Right? Over time, it really creates this community of bacteria. And you have a variety of other bacteria cells that come in. So you've got these red guys, you have these white guys, the yellow and the purple here. I think that's just extra part of the kind of structural components of the biofilm. And those structural components are going to be made by and secreted by the different bacteria cells that are within that community. So that's a biofilm. All right, the capsule is bound more tightly to the cell. It's going to be dense. It's going to be thicker than a slime layer. And you can see it by negative staining. So these cells here, this is the bacteria cell, that red dot in the middle. And then the yellow halo around it, that is going to be the thickness of the capsule. So you can see how thick that capsule is relative to the actual size of the bacteria. When you look at it on a petri dish, it looks snotty. Right? It's got that mucoid appearance to it. And encapsulated bacteria cells generally have a greater pathogenicity. Again, because it's harder for the phagocytes to grab on and to kill it, and so it goes unchecked and replicates to high numbers to cause infection. Alright, so quickly, just as a review, right, what are the two types of glycocalyxes? Mentioned one very well, not so much the other. Alright, slime layer and capsule. Alright, which one is more tight? Alright, so which one is more dense? The answer would be the capsule. You can cue the Jeopardy song here if you wanted to. Oh, and alright, so what is one of the 3 clinical significant purposes of a flagella? So I didn't mention this. Got it on my notes, but didn't look at my notes. Alright, so you've got movement of the bacteria cell. Why is that important? Alright, one, it allows for an opportunistic infection to arise. And let's use E. coli as a quick example. E. coli has a high population within our intestinal tract. It has a high presence within fecal material, and that fecal material is going to have to leave our body. And let's just be real, it's not always a clean escape when it leaves the body. So the surface of our skin can, in some areas, have E. coli on the surface. Now, E. coli is a gram-negative organism, and it possesses flagella. So what can it use? It can use that flagella to move across the surface of our skin and gain access to areas that it shouldn't be, such as the urinary tract system. So, the organism that was in the GI tract, perfect place for it, gets into the bladder or the urinary tract system, not cool, and now it is an opportunistic infection, being that urinary tract infection. And it also allows it to move, which simply allows it to escape from the immune system. And then finally, if it can move, it can develop into a systemic infection, meaning it can go anywhere in the body. It can go everywhere in the body, and it not just be something local, but rather systemic.